have mad minutes about the poster, which are right over there. It's going to be quick and very exciting because you'll get to hear about everyone's research. So pay attention, remember where everyone is, and enjoy poster session. Okay, we're starting, and I'll take it's mad minute. It's not mad 30 seconds, and it's not mad 2 minutes, it's mad minute. Okay, I'm just saying. Okay, starting, bye.
Hi, I'm Dave. My poster will be about, we take data simulation, which is the machine learning-like tool that they use to forecast hurricanes or get Cassini to fly into Saturn. And we leverage physiologic information, such as can be captured in endocrine models. And we marry it with data, and then we try to forecast what's going to happen to people or infer measurable and unmeasurable parameters that we can use to phenotype patients. So we'll have sort of figures of how that all works, and I can explain what it's about. That was a lot less than a minute. I'm sorry.
Hello everyone, my name is Janet Woolen, and I'm part of the amazing Sue Bawkins lab. Our lab is focused on reducing health disparities in our society. One of the projects that we're focusing on is helping vulnerable individuals with low health literacy and limited English abilities to understand genetic test results. If you'd like to learn more, come to poster 2. I can keep going, but I don't know how long.
Every year it gets shorter. Okay.
This was going to be one word. No. Hi, I'm Gal. I'm a third-year PhD student working with Noemie. Yes, we have the same haircut right now. The poster is not about that. The poster is showcasing our progress on a patient record visualization tool. It exhibits patient problems and their dynamics over time. Currently, we're leveraging the correlated topic model, and we have a fancy interactive visualization using D3. So come on by and hear about our findings from our user study and our next steps.
Hi, everyone. My name is Molly. I'm a fourth-year PhD student in Dr. Elhadad's lab. And my research focuses on characterizing the patient experience of disease. Specifically, I look at endometriosis. So to do this, my research team created Fendo, which is a smartphone app that allows for tracking a customizable set of signs and symptoms of the disease, as well as provides a standardized clinical questionnaire through an electronic portal in the app. And we find that the data collected through Fendo is generally congruent with existing knowledge about the disease. But we also looked at the same users reporting through Fendo and also reporting through the standardized clinical questionnaire. And we find that there's a greater range and granularity of symptoms reported in Fendo as compared to the standardized clinical questionnaire. We also looked at dynamic disease variation and found day-to-day and cycle-to-cycle variation across all disease dimensions. So our results show that 1, app-based self-report is valid, and 2, that the temporal aspects of endometriosis.
Hey everybody, my name is Victor. I am a second-year PhD student working with Noemie. So my poster is on automated method for phenotype discovery. So methods for automatic discovery of phenotypes are considered fundamental for tailoring medical interventions. However, a large proportion of chronic diseases and morbidity and mortality are attributed to adverse health behaviors, which are most responsive to behavioral interventions as opposed to medical interventions. And so what we do is we apply automatic methods for phenotype discovery to survey data that probes health behaviors, their mediators, and their moderators. So for more, come check out our poster. It's poster 5, I think. Thanks.
Hello everyone, I'm Inigo. I'm a postdoc working with Noemie on EHR phenotyping. And we're trying to basically learn computational models of disease at large scale and in an unsupervised manner. And this raises several challenges. One is how to combine patient specifics with population level commonalities. Another one is how many phenotypes to expect in a population. And maybe more importantly, how these learned phenotypes extend or are applicable to unseen patients. Do we need more phenotypes? Do we have to learn more or less? So to answer these questions, we're using machine learning and statistics methods. In particular, we're using non-parametric Bayesian statistics, which is a fancy name for some methods that statisticians have been working on for many years. So we have some preliminary results. If you want to learn more about our results or about the methods, just stop by. It's poster number 6. Thanks.
Hi, I'm Ken Smith. I work with RS Florados. So for the last several years, we've been working on a project with the National Cancer Institute to help develop a web-based application for sharing detailed research findings among a group of cancer networks, a network of cancer groups. So this is the Cancer Target Detection and Development Network, and it focuses on characterizing functional genomic alterations, screening for active compounds, and new biomarkers. So what the system does is it allows you, the researcher, to formulate concise descriptions of research findings, building upon controlled vocabulary for key items like genes and compounds, cell lines, and also for the roles of these in the statement. And then it also provides for attaching any kind of experimental evidence to the statement. So the idea is for the researchers to be able to share information.
Sorry, I'm looking for Ben Coomer. Are you here? Oh, no. Okay, we're going to PJ. Oh. Sorry.
Okay, it's PJ. I'm an internal medicine specialist and first-year clinical informatics fellow. My personal project has been about developing a framework for apps, clinical decision support apps for clinicians. Clinical decision support could be simply just telling physicians to do this and do not this. But as a clinician, what I think is that the decision support should be combined with education, because there's clinicians who want to seek a reasoning behind any decisions. So I try to build a frameworks which allows for building apps, which tells physicians what to know at the point of care rather than telling physicians do this and do not this. So as a trial, I happen to work with a psychiatrist at Harvard to build an app for a psychiatrist, pretty much transferring a clinical psychiatrist guidelines into app. So you can come and see. It's available in the App Store. Right. It's free.
I'm Richard Friedman. My work describes a collaboration with the Wang, Su, Honig, and Califano labs, in which we discovered the cell of origin of pancreatic cancer. We find that pancreatic acinar cells which express DCLK1 are tumorigenic, provided that they also have KRAS G12D mutations and are subject to inflammation. We also propose a molecular mechanism about how this takes place. I have a half a minute left for questions.
Okay, George representing Sunny Shang, recently promoted to Associate Research Scientist, more recently promoted to Mom. She gives her regards from she and her daughter Valerie, born a couple days ago. Sunny's working on phenotyping, joining the eMERGE project with the Odyssey project. eMERGE project is famous for practical solutions to taking electronic health record data, turning it into usable phenotypes. The problem is, once you define a phenotype in one place, it takes a year or two for all the other 10 sites to convert it to their database to do it there, so to do it at their location. So Odyssey is bringing in the standards for that, and the work has a lot of knowledge engineering components.
That's it. Thank you.
Hi everyone, my name is Matt Levine. I already pressed start. So this is how you avoid shame from Noemie, as you press start before her. So I'm presenting work that I've done with George Ripsack and Dave Albers, doing time series analysis on EHR data for detecting physiologic effects of drugs. And you can imagine this is a useful thing to find for a number of reasons. One of them that we look at or think about is finding adverse effects of drugs. And specifically, we look at a simple class of time series models, just lagged linear regressions, and study different aspects of how changing those models can impact the predictive performance. We have 2 gold standards, and we compare to them, and I have 20 seconds. Great. Come see the poster, please.
My name is Daniel Feller, second-year PhD student. My poster is on the prediction of HIV using clinical notes and structured data in the EHR. Worked with a group of HIV clinicians at Columbia to show that a simple language model using unigrams can help predict HIV from structured records.
Hey everyone, I'm Elliot Mitchell. I'm also a second-year PhD student in Lena Mamakina's lab. I'm going to ask for some help from a friend. Hey Siri, will you explain what my poster is about?
Sounds a little gimmicky, but okay, let me check. Elliot will present a poster on preliminary research about the applications of voice interfaces in healthcare. You might know about voice interfaces like my BFF Alexa, or my arch nemesis OK Google. Stop by if you want to hear about a prototype voice-based application to improve diabetes self-management by providing personalized nutrition recommendations.
Thanks.
Hi everyone, I'm Sylvia Cho, working with Dr. Karthik Natarajan. My poster is about mapping concepts from a transplant registry to concepts in the OMA vocabulary. Currently, the CMS monitors transplant outcomes based on data from a transplant registry, but to conduct better outcomes analysis, we need to integrate multiple data sources such as EHR and claims data with the transplant registry. And this can be achieved by leveraging the OMAP CDM, which standardizes the structure encoding system. As a first step to this, we evaluated the content coverage of the OMAP vocabulary towards the concepts in the transplant registry. And two annotators individually mapped 3,500 unique source concepts to OMAP concepts using a tool named Usagi. And we were able to map about half of these concepts with good agreement. Please come see our poster if you would like to hear more about it. Thank you.
Hey everybody, my name is Amelia Averett. I work with Dr. Adler-Parat. Today I'm presenting a poster called the Noisy or Risk Allocation, or NORA model. And this model, under a set of simplifying assumptions, a single binary outcome, and a set of binary features, will give you the risks associated with each exposure, which helps determine the true causal factor. I think I'm poster number 16, so feel free to stop by and talk about it some more.
Hey everyone, I'm Alex. I'm a fourth year in Yufeng Shen's lab. So broadly, our lab is interested in studying genetic variants in the context of developmental disorders. And recently, there's been a lot of work implicating de novo mutations, or mutations in children, but not their parents, in these types of diseases. And so for our poster, my current project is developing a computational method to identify mosaic mutations from blood whole exome sequencing data from congenital heart disease and autism patients. So if this is something that interests you, come check out my poster.
Hi everyone, I'm Hong Jian. I'm from Dr. Yu Fengshen's lab. Today I'm presenting actually for Alex K because he's on his honeymoon and enjoy his holidays. So the work we are working on is trying to understand the genetic architecture in the non-coding region, because coding region only explain about like 20% of the developmental disorder disease. So we develop a computational method and do simulation to find something interesting in the non-coding region. So that's it.
Hi everyone, I'm Joe Romano. I'm a PhD student in Nick Tatinetti's lab, and I study animal venoms, so specifically animal venoms that have therapeutic effects on the human body. My poster is about two different studies. The first of them is something we call Venom-seq, where we take 25 different venoms and we expose them to human cell lines. We then do RNA-seq and generate differential expression profiles so we can look for potentially therapeutic effects that they have. The other study that we also report on there is Venom-KB, which is a way that we can then report the results in a standardized fashion using semantic knowledge structures that people can access using computational methods. So I believe I'm poster 19, so come check it out. Thanks.
Hi everyone, I'm Phyllis Thungaraj and I work in Nicholas Tatinetti's lab. I'm, I guess, now a fifth year MD PhD student. Sorry. And it's confusing. Anyway, so if you're curious about phenotyping in the EHR and or stroke, you should come to my poster. Basically, I've developed and analyzed 50 different models comparing curated data sets versus those using diagnosis codes to determine which training sets and models can most accurately predict and identify people in the EHR with stroke. So come to poster 20 if you're curious.
All right. Hi, everyone. I'm Alex. I'm in Nick Tatney's lab. I'm a third-year PhD student, and I'm not going to talk to you about my research with him. I did a project with Tuli Lapalainen at the Genome Center during my first year, and I ended up developing a tool called Hyper for high performance and robustness to analyze the data from CRISPR gene editing experiments. We compared it to the best competitor called CRISPR-Eso, and we're doing better in accuracy, over 98, 99% accuracy of classification of the reads. And we're also much faster, between 5 and 6 times faster. So come check my poster, and please don't ask me anything about genetics.
Hi, everybody. I'm Lisa. I'm also an MD PhD student in some year, and I work for Dr. David Vaudry. Our poster is about models that predict hospital readmission risk, and these models have garnered a lot of attention recently because readmission rates can affect Medicare and Medicaid reimbursements. And so we conducted a national survey of researchers and healthcare organizations to understand how these models are actually being implemented on the ground, as well as barriers to developing and implementing quality models. So come check it out. Should be interesting.
Hi, everyone. My name is Fernanda Polubrejnov. I work with David Vaudry as well. And my poster today is, our work focuses on patient-provided data. And today the poster is about a method that we developed in collaboration with Nick Tatnetti's lab that uses emergency contact information to extract familiar relationships from observational databases. We validated these relationships using genetic data, and then we used them to compute heritability of 500 different traits. So if you're interested, come check it out.
Nature ran its own course, and over time developed sophisticated mechanisms that evolve languages that seem to be governing many of the biological processes. And this can be observed in many social interactions among sentient beings, down to genetic and protein sequences on microscopic level. So I like to suggest that there's no exceptions in the symbolism when it comes to complex disease processes and human behaviors surrounding these processes. My name is Barnett Chu, and I'm currently working on a neurolinguistic-based framework that helps to decipher those hidden messages embodied within EHR and healthcare systems. The first application will be in the disease subtype discovery, that through analyzing temporal sequences of medical codes, which I refer to as medical coding sequences, drawing from the analogy of genomic literature.
Hi, I'm Andrew Goldstein. I'm a third-year postdoc with Chunhua Wang. Thank you, Patrick, for warming up the audience to science being both broken and awesome. So, I'm a primary care doctor during med school and residency. We do journal clubs. We critically appraise the scientific literature to better apply it, think about future research, find bad science, find good science. All this conversation in person, all the commentary and journal comments that are published kind of gets lost and doesn't get used. So think of it as like an information loss issue that's very prevalent and unfortunately underappreciated. So we conducted a scoping review of what happens to the critical appraisal of clinical trials by informed stakeholders. Does that information get captured, managed, organized, used? And if not, what are the gaps? And we published that in JAMIA, and we have a poster at poster booth 25 if you want to learn the punchline.
Hi, everyone. I'm Tan. I'm a second-year PhD student working in the Dr. Chunhua Wang's lab as well. Today, my poster represents an information extraction system to develop for the eligibility criteria to parse and standardize it into the ALMA common data model. So if you're interested in the ALMA, NLP, or knowledge representation, you can find me at poster 27. I'm really happy to discuss this with you. Thanks.
Hello everyone, my name is Chi Yuan. I'm a second year computer science PhD student. I'm working with Chenghua. My poster will be a criteria to query project. It presents a pipeline for translating free text criteria to OMOP common data model-based SQL query. It can help people who don't have any computational skills to create their SQL query, and it can make their focus their energy on their studies and can help them to do their criteria design better. If you are interested in this project, please come to my poster. Thank you.
Hi, everyone. My name is Ben Coomer. I'm a second year clinical informatics fellow. I just put this together. I'm also a stroke neurologist, and I spent a good part of my first year working on the Mobile Stroke Unit, which is a novel NYP-led initiative that allows stroke treatment, including CT scan and a clot-busting medication, to be delivered in the field, i.e. in a very large specialized ambulance. And my poster is on the integration of NYP's information systems into the ambulance itself, sort of treating the ambulance as an extension of the emergency department. So we sort of included the registration system, the lab system, and the EHR, which is all scripts, at least for now, into the ambulance. So if you're interested in sort of applied information systems and things of that nature, stop by my poster. And our times are actually really good. We've showed that we can actually treat people 30 minutes shorter.
It controls.
So I wrote down what I have to say, otherwise I know I'll go to 5 minutes. So I'm Rita Kukafka, and I work with a group that is very interdisciplinary, interested in developing an infrastructure for implementation of personalized medicine and primary care practice settings. Following risk stratification of individual patients, we provide decision support for both providers and for patients. Our decisions that we focus on are preference sensitive, that is, we incorporate the best available evidence, plus patient preferences, and we study outcomes, which include clinical outcomes, use of services, and decision quality metrics, patient-reported outcomes. For example, was the decision informed, and what was the process? Was it a shared decision-making process? We have funded studies through the NCI and through ACS. We study genetic testing decision-making, and we study chemo prevention as well. And come visit us. See, I wrote it.
Works. Hello, I'm Draško. I work with Lina Mamikina. So if you ask patients what they think about online discussion forums, they will tell you they're a mess. So what I'm trying to do is basically to understand this mess and understand how people make sense of this mess. And based on that, develop tools that will support this sense-making process. So today I will be presenting a system called the Explorer that supports this sense-making process by recommending or suggesting relevant posts in the discussion based on one that's selected as a post of interest. So you can stop by number 30. Thanks.
Thank you, everyone. See you at the posters.